welcome to the Saurya Show. So this is the tutorial series in the Saurya Show where we will be discussing the topics of medical science in a very short period of time so that you can get maximum output. So watch the video until last. So today's topic is white blood cells, right? So and its type. So let's go. The types include neutrophils, right? So it includes neutrophils, then it includes eosinophils and basophils, right? So neutrophils are the cells which is in maximum quantity in the cell, right, in the WBCs. So the percentage is 54 to 62%, right? And the eosinophils, the quantity is 1 to 3%, and basophil is the least, that is only 1%, right? And we also have to know the normal WBC count, which is 4,000 to 10,000 per mu-L, right? And we also need to know one clinical condition, right? So this is a clinical condition that we need to know, and that is increased amount of WBCs or leukocytosis. So in the conditions like infection, some kind of bacterial infection or any kind of infection, and in also in the cases of cancer, malignancy, there is increased WBC or leukocytosis is seen, right? So now let's go into more detail about the neutrophils, monocytes and macrophages, right? So neutrophil is a multi-lobed, right? It's a multi-lobed, you can see 3 lobes, 1 2 3, right? So it is multi-lobed, it has multi-lobed nucleus, and there is small pink granules. So you can see the pink granules there, because in H&E stain it will stain into pink, right? And the nucleus will be stained into blue. So that's the identifying feature, and the function is phagocytosis and activation of bactericidal mechanism. And the next one we have monocyte. It is also found in bloodstream, and the characteristic feature is it has kidney bean shaped nucleus, and the function is phagocytosis, right? And differentiate into tissue macrophages, right? So monocyte will be differentiated into tissue macrophages. And the next one that we have is macrophage, right? The next one that we have is macrophage, and the macrophage is not found in bloodstream but is found in tissues, right? And the function is again phagocytosis and secretion of cytokines. And now next one is eosinophils. You can see, right, eosinophils has bilobar eosinophilia, right? Where is the okay, so hypereosinophilia. This is the very important clinical condition, and there is increased eosinophil in case of neoplasm, right? In case of asthma, in case of allergic process, and in case of collagen and vascular diseases. And now we have basophils, right? We have basophils. Basophils is found in bloodstream. It has bilobed nucleus but large blue granules, right? So we can see the blue granules here, right? So these are the blue granules. So we also know that in H&E stain, basophil retain the color of blue stain, right? So that's why it appears to be blue, and the function is not phagocytosis in case of basophils, right? So the function is it release pharmacologically active substance during allergic responses, right? And now we have mast cells, right? So mast cells is very important, it's really important cells, and it is located into tissue, mucosa, and even in epithelium, right? And we can identify because of its small nucleus, right, and large blue granules. You can see the granules here, right? And the function is the granules, these granules release histamine, right, during allergic conditions or allergic responses. And we also have to know some of the symptom of mastocytosis, that is increased quantity of mast cells, and the symptoms such as itching, flushing, abdominal cramps are the symptoms in which mast cells is increased, or histamine is more amount of histamine is released. And one very important point to be noted is it is involved in type 1 hypersensitivity reaction. So this is very important point to be noted. Now we have the mature myeloid and lymphoid cells, right? So now let's get to know about the mature myeloid, right? So the mature myeloid and lymphoid cell. So dendritic cells, right? It is found in epithelia tissues and the Langerhans cells, right? So when the dendritic cells are found in epithelia, it is called as Langerhans cells. So this is also one of the immune mechanism, and we can identify it because of its long cytoplasmic arms, right? And it expresses MHC class 2, right? And the function is antigen capture, transport, and presentation. So dendritic cells capture the antigen, it transports, and it also helps in antigen presentation, right? So then the next one that we have is lymphocyte. So lymphocyte is found in bloodstream, right? It's found in the quantity is 1000 to 4000 per ml, and we can see the lymph nodes, spleen, submucosa, and epithelia, right? And how can we identify lymphocyte? By large dark nucleus and small cytoplasm, very small cytoplasm, right? But there is large dark nucleus is large, cytoplasm is very less. And the functions of lymphocyte are it consists of B cells, right, T cells, Th cells, right? So B cells produce antibody, right? And from where B cells are matured, so the B cells are matured into bone marrow, and T helper cells are matured into thymus. So B for bone marrow, T for thymus. So B cells in bone marrow and T helper cells in thymus, and they both regulate the immune responses. And cytotoxic T cell kills the altered or infected cells. Now the next one that we have is plasma cells. So plasma cells are found in lymph node, it is found in spleen, it is found as mucosal associated lymphoid tissues and bone marrow. So how can we identify it? By its dark nucleus, right? Intensely dark staining nucleus, and obviously there is Golgi apparatus, and by this we can find it as a plasma cell. And the function is the end cell of B cell differentiation, and it produces antibody. So this was about today's class. Hope you like it, and if you like the video, please comment down below what kind of videos you want to see, what kind of topics you want to me to cover, and please subscribe to the channel also. Thank you, have a great day.